Good morning, dear students of class 9. Today we will begin with the new unit and a new chapter. Unit number 4, Diversity in Living Organisms, and chapter number 9, Economic Importance of Bacteria and Fungi. Okay, so mainly in the new reduced syllabus, chapter number 8, 5 Kingdom Classification has been removed. So we have to go with the chapter number 9, Economic Importance of Bacteria and Fungi. Clear? So we are dealing with the unit number 4, Diversity in Living Organisms, with the chapter number 9, Economic Importance of Bacteria and Fungi or Fungi. Alright, so what do you mean by bacteria actually? Because we are used to hear the word bacteria, viruses, even we have learned different types of diseases which have been caused by the bacteria, right? When we're talking about human health and diseases, which has been removed from the syllabus, right? But we have done or already covered those name of the diseases which have been caused by the bacteria. Okay, so what do you mean by bacteria? Actually, bacteria are the primitive organisms. They are unicellular, which are made up of single cell. All right, which are also called the prokaryotes. All right, they are unicellular organisms which are called the prokaryotes. Primitive means the earliest form. Clear? They were unicellular, which were made up of single cells. And the most important thing is that these bacteria, they are without the nuclear membrane. Clear? It's also called they don't have a well-defined nucleus. Eukaryotes used to have a well-defined nucleus, meaning that the nucleus is covered by the nuclear membrane, and our genetic material, that is DNA, is within the nucleus. Clear? But in case of bacteria, in case of prokaryotes, a well-defined nucleus is not present. That means the nucleus is without the nuclear membrane. So therefore the genetic material in case of bacteria, alright, that is the single chromosomes, let's say, it's usually present in the center of the cell. Clear? It will be free floating in the cytoplasms. In our case, it will be within the nucleus because it is enclosed by the nuclear membrane. But in bacteria, there is no nuclear membrane, so the genetic material will be freely floating in the cell cytoplasm. Clear? So therefore, we can say that in case of human being, all right, in our case, I'm giving examples. In case of human beings, in our body, our DNA is nicely packed within the nucleus. Clear? Where it has to accommodate within the small nucleus. But in case of bacteria, since there is no nuclear membrane, right? So therefore, the genetic material in bacteria will be enclosed in a form known as nucleoid. 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 Clear? This is the brief information for you. Now there are several thousands known species of bacteria which occur in air, water, soil, food. Okay, and even inside the body of living organisms also. And in short, particular we can say everywhere. In our body, there are hundreds and thousands of bacteria that occurs in the mouth, in the small intestine, in the skin also. Clear? And even there are few beneficial bacteria. All the bacteria are not harmful, but there are those bacteria which are beneficial also. Example, I can say E. coli. Okay, that will help in synthesis of certain vitamins in our body. Okay, they will prevent the growth of other bacteria also. So there are not only the beneficial, sorry, not only the harmful bacteria that cause different types of diseases, but there are beneficial bacteria also. Okay, now let's come back to the size of the bacteria. So when we consider the size of the bacteria, the average size will be around 2 micrometer long and 0.5 micrometer thick. So 1 micrometer means 1,000 of a millimeter. Shape wise, we have got four types of bacteria. Cocci or coccus. Okay, the shape is spherical. Bacilli or bacillus, the shape is rod shaped. Spirilla, spiral or twisted bacteria. And Vibrio, the comma shaped bacteria. So remember this one. So based on this shape, so according to the shape, there are four types of bacteria. Cocci, shape is spherical. Then bacilli, the shape is rod shaped. Spirilla is twisted or spiral. Okay. Then vibrio, comma shaped bacteria. So these are the different types of bacteria according to their shape. Now, many bacteria, they occur in single. Okay, they live in singly means they occur in single. Others occur in pair, in double. Okay, like diplococci, or in long chain called streptococci, or in clusters called staphylococci. Now we'll see the diagram. So you can see over here. So this is the single bacteria called monococcus. Okay, when they occur in pair, diplococcus, they can occur in cluster called sarcina. Okay, or even they can occur in mm, long chains called streptococcus. Okay, so these are the different types of bacteria. Okay, based on their occurrence, either in single, either double, or either in a group, or in a cluster, or in a chain. Okay, now next one we'll go with the structure of bacteria. All right, this is just the brief introduction of bacteria which you have to remember. All right, afterwards we'll go with the topic that is there in the syllabus. But before going into the topic that is in the syllabus, a brief idea about bacteria must be known to every one of you. Okay, so kindly listen very carefully. All right, we have just finished about the shapes of the bacteria. Now structures, when you consider the structure of the bacteria, it contains living substance protoplasm. So over here the structures is known as protoplasm. Okay, or the protoplast, which lies within the cell membrane. So you can see over here the cell membrane. Okay, so this entire structures over here is the protoplast or the protoplasm. Now lying within the cell membrane, we will find the protoplast, which is surrounded by the non-stiff cell wall. You can see the cell wall over here. So we have got the cell wall, then cell membrane, then we will find the protoplast. Okay. Now the cell wall is made up of peptidoglycan, a polymer of sugar and proteins. We can see. Okay, peptidoglycan means a polymer of sugar and proteins or amino acids. Now in case of bacteria, there is no well-defined nucleus that we know because there is no nuclear membrane. But chromatin material, that is DNA, is present in the central region. Okay, this is very important. So you can see over here the bacterial chromosome. So this intact part, let me make clear. So this part, the central structure, is the bacterial chromosome. Okay, there is no well-defined nucleus, there is no nuclear membrane, so these bacterial chromosomes are present in the center of the cell. Okay, then we'll find an extra chromosomal circular DNA molecule in bacteria also. One more time, extra circular chromosomal DNA. Okay, that extra circular chromosomal DNA except these bacterial chromosomes also. All right, we have got extra chromosomal circular DNA molecule in bacteria, which is called the plasmid. So it is the extra chromosomal circular DNA molecule present in bacteria. So in bacteria, we'll find the bacterial chromosome. Fine. Along with the bacterial chromosome, we'll also get plasmid. Clear? Okay, in higher class, you will learn that these plasmids are used in a technology called biotechnology for the formation of different types of substances, okay, like medicines, like vaccines, all those things. Clearly we learn in higher classes. So what I learned till now, we are dealing with the structure of bacteria. We learned that in bacteria there is no well-defined nucleus, right? So going to that, the bacterial chromosomes are present in the central regions. Then we'll find the extra chromosomal circular DNA also, which is called the plasmids. Then we'll find the living protoplast inside the cell membrane, and cell membrane is surrounded by the cell wall, which is made of peptidoglycan, and peptidoglycan is mainly a polymer of amino acids and sugar. Okay. The cytoplasmic part contains vacuoles, granules, and in some cases chlorophyll as waste. Chlorophyll in case of blue-green algae, also called cyanobacteria. Okay. Then in few cases, in certain bacteria, we will find the protective layer, also called the capsules. Then bacteria are able to move by the help of flagellum or flagella. Okay, they can move from one place to another place by the help of this flagella. Clear? Then nutritions, we have got heterotrophic mode of nutritions because they are not able to prepare their own food by themselves. So we have got heterotrophic mode of nutrition in bacteria. And under heterotrophic mode of nutrition, we have got two types, saprotrophic and parasitic. Saprotrophic bacteria means those that will obtain the nourishment, those that will obtain the food materials from the dead and decaying organisms. And parasitic means those that will obtain the nourishment, the food particles from the other living organisms which are known as the host. Okay, so mode of nutrition, we have got heterotrophic mode of nutrition, because most of the bacteria they have no chlorophyll. Okay, those who have got chlorophyll, they are called photoautotropic bacteria. Clear? But those who are not able to prepare their own food, they are called heterotrophic bacteria. And under the heterotrophic bacteria, we have got saprophytic bacteria and we have got parasitic bacteria. Saprotrophic means those will obtain the nourishment from the dead and decaying organisms, simple from dead bodies of living organisms, sorry, of dead organisms. And next one is parasitic, which will obtain the nourishment, their food from the other living organisms, which are called the host. And in higher classes, you will learn one more thing under the types of nutrition under heterotrophic, that is called symbiotic bacteria. Okay, which you learn in case of the nitrogen fixation. Clear? So symbiotic means where the bacteria will live in close association with other organisms, where both of them will be mutually benefited. Kindly remember this thing. Okay, now let's go into the respiration. So respirations will be aerobic, which will take place in the presence of oxygens, and anaerobic, where there is no need of oxygen. Okay, and this anaerobic bacteria will be killed if they are exposed to air. And similarly, aerobic bacteria, which will survive only in presence of oxygen. If oxygen supply will be cut, if there will be no oxygen, then those aerobic bacteria will also die. Okay, now let's come to reproduction. Reproduction in bacteria is asexual, is involves single parental. It takes place without the formation of gamete and without the fusion of gamete. So reproduction in bacteria is simply asexual, which takes place by means of fission. Fission means to break. Clear? So in this case, what I mean is that the circular DNA replicates. So you can see in the figure, this one is the circular DNA. Okay, the circular DNA replicates means it will form its own carbon copy. Alright, so the circular DNA will replicate. So you can see over here. So first copy and the second copy here. Okay, so original DNA you can see over here, and this original DNA will replicate to form the new copy of DNA. First, the circular DNA will replicate. Then the cell start constricting. Can you see over here the structures? Let me make it clear. So afterwards, what I mean that the cell start constricting. Can you see over here? A furrow will develop at the center of the cell. The cell start constricting at the middle, dividing the cell into two. Okay, so this furrow or this constriction will start growing until and unless it will finally divide the parental cell into two daughter cells. Clear? So what I mean is that in reproduction fission, first of all, the circular DNA of bacteria, it will replicate. All right, it will form the copy DNA. Then at the center of the cell, a constriction will appear. That constriction will grow, will become more and more deep. Okay, until unless the parental cell will divide into the two daughter cells. So this is called fission in case of bacteria, which is a mode of asexual reproduction. Clear? Okay. Now we'll go into the topic. Topic. We'll talk about the useful role of bacteria in medicines. Alright, so now these things are there in syllabus. Clear? So now we'll go with the topic that are there in the syllabus. Okay, now till now, whatever we have done is just for your basic knowledge regarding the bacteria. Okay, so this is the syllabus which we have to remember. So let's talk about the role of bacteria in the medicines, because I've been telling you that not all the bacteria they are harmful, but there are certain bacteria which are also beneficial. Okay, so now we are going to talk about those bacteria which are beneficial. So first of all, we'll talk about the role of bacteria in medicines. So bacteria are mainly used in the production of antibiotics, they are used in the production of serums, and they are used in the production of vaccines. Alright, so first one we'll talk about the role of bacteria in the production of antibiotics. Okay, so today we know about dozens of antibiotics which are being widely used in different types of diseases, in different types of infections. For example, like streptomycin, right? We can learn the thing. Then we have got chloromycetin, we have got erythromycetin, we have got tetracycline, we have got penicillin. These are the different types of antibiotics which are now being used for the treatment of different types of infections. Clear? So penicillin, I think I heard this term, the antibiotic penicillin. Penicillin was the first antibiotic which was produced from a fungus, Penicillium chrysogenum. It's also called Penicillium notatum, right? So this penicillin is the first antibiotic which is being produced from the fungus Penicillium chrysogenum, or simply it used to be known as Penicillium notatum. Okay, so how was this penicillin discovered? Who discovered penicillin? Sir Alexander Fleming in the year 929 he discovered penicillin, the first antibiotic. So he was working on a bacterial culture. Okay, let's say we have got the two culture. In the two culture plate, he was observing the growth of the bacteria. So in one of the culture, bacteria was growing normally, but in the other culture, the growth of the bacteria or the multiplication of bacteria was stopped. Okay, so when Fleming observed the regions behind the cessations or the stoppage of the multiplication of bacteria in the culture, he noted that a certain mold, mold, mold of fungus called Penicillium chrysogenum was present in that culture. Okay, the culture where there was no growth of bacteria, in that culture, a mold of fungus Penicillium chrysogenum was growing. It was formerly known as Penicillium notatum. So don't get confused between Penicillium chrysogenum and Penicillium notatum. Clear? So that mold of Penicillium chrysogenum or Penicillium notatum was growing in the culture plate where there was no growth of bacteria. And he found out that these mold of bacteria, right, used to produce a substance, and that substance was suppressing the growth of the bacteria. And later on, he named this substance, which was produced by Penicillium chrysogenum, is penicillin. So penicillin is the substance which is produced by Penicillium chrysogenum, which was inhibiting, which was stopping the growth of bacteria. So penicillium is the product of a mold fungus. Remember this thing. Penicillium is the product of the fungus, not that of the bacteria. That means penicillin is produced by the fungus Penicillium chrysogenum, formerly known as Penicillium notatum. It is not produced by the bacteria. So penicillin is the first antibiotic which was produced by Sir Alexander Fleming. The second antibiotic we can say, or the antibiotic streptomycin, was produced by Salman Waxman. Okay, it was produced from Streptomyces gracius. Okay, and it is used for the treatment of tuberculosis. You can look at the table over there. Penicillin is used in the treatment of tetanus, diphtheria. Streptomycin is used in the treatment of tuberculosis. Clear? Similarly, chlorotetracycline is obtained from Streptomyces aureofaciens and is used in the treatment of typhoid. And erythromycin, the antibiotic, is obtained from Saccharopolyspora erythrea, and it's used for the treatment of rickettsial fevers. Clear? So these are the name of the few antibiotics, the source organisms, and the disease for which they are being used. Okay, now we'll talk about the criteria for good antibiotic and the other uses of antibiotics. So what are the criteria for the antibiotic to be good? It should be able to kill a variety of disease causing microorganisms. Okay, that means it must have broad spectrum. It must kill a large number of disease causing microorganisms. It should not produce any kind of side effects when we are using it, and it should not kill the normal bacteria of the host, because we used to harbor a lot of bacteria. Among them, most of them are beneficial. So whenever we are using those antibiotic, those beneficial bacteria should not be killed by those antibiotic. Clear? Other uses, it can be used as food preservatives for preserving fish meat. Okay, then for treating animal feed, and for controlling the plant pathogens. So these are the few examples where we can use the antibiotics. Sorry. Next one, serums, for producing serums. Now what is serums? Now serum actually means the blood plasma from which the fibrinogen has been removed. What is fibrinogen? It is mainly used in the process of blood clotting. Okay. So those blood plasma from which the fibrinogen has been removed, they are called the serum, and it contains antibodies, or we can say antitoxins. Okay, now what do you mean by toxins? Toxins and antitoxins we have already learned in the chapter It's to Health, or Human Health and Diseases. So we are familiar with this topic. So toxins are the harmful poisonous substances or the chemical substances which are produced by the pathogenic organisms like bacteria, viruses. Okay, and antitoxin is a substance produced in the animal body which react with the poison produced by the invading germ. Antitoxin means they will work against the toxins. Clear? So all these things we have done already in the unit human diseases, right? It's to health also. So already you are familiar with this term. So I'm not going in details about toxins and antitoxins. So how can we prepare the serum? Sorry for that. For preparing serum, a small dose of bacterial toxin sorry, a small dose of bacterial toxin is introduced into the blood of a healthy animal. So what we are doing whenever we are trying to prepare the serum, so what we are doing is that a small dose of bacterial toxin is being injected into a healthy organism such as cow or horse. Okay, so when we inject the bacterial toxin, let's say into the body of a horse, the body of the horse will start producing antitoxin. So antitoxins are produced in order to neutralize the effect of toxins. Clear? So after several injection of the same toxins, all right, the blood of such animals will be taken out. It will be chilled, and when those blood of the animals will be chilled, it will produce a clear extra-colored liquid which is called the serum. Clear? Simply, what we are doing, we are putting the bacterial toxin into the body of a healthy animal such as cow or horse. Let's say, for example, horse. Now, when we are putting the bacterial toxin into the body of the horse, within the body of the horse, there will be formation of antitoxin. Antitoxin will work against the toxins produced by the bacteria. Okay, so whenever we are administering or several injection of toxins into the body of the horse, the body of the horse will develop antitoxins. Now from the body of the horse the blood will be taken out. Okay, that blood will be kept for chilling. On chilling, it produces a clear extra colored liquid which separates off from the blood clot. That extra colored liquid is known as the serum. Okay, even for the snake bite, we used to take this serum or the anti-venom which are produced in the body of other animals like horse. The hot word anti-venom or anti-serum, this we have already discussed, right? When we were talking about immunity. Okay, so next one, genetic engineering. All right, the modern advancement in the field of biology, biotechnology, or genetic engineering has also made it possible to introduce the human gene into certain bacteria like Escherichia coli or E. coli. All right, in this way, you can make different types of useful products for the human being. For example, the hormone insulin, which is being used for the treatment of diabetes, was first produced by the help of genetic engineering using E. coli or Escherichia coli. And the name of that insulin was known as inulin, inulin, inulin. Okay, so hormone insulin was also being produced by the process of genetic engineering by using or by using the bacterium called Escherichia coli or E. coli. Okay, now a few serum compounds which are produced genetically by genetic engineering. Okay, by modified bacteria are blood clotting factor 8 for the treatment of hemophilia A and factor 9 for the treatment of hemophilia B. So these blood clotting factor 8 and factor 9 are being produced by the process of genetic engineering by genetically modifying the bacteria. And simply we are using which bacteria? E. coli or Escherichia coli. The next one, vaccines. You are also familiar with the term vaccines, a preparation which consists of dead or weakened germs. Remember, in vaccines, we are usually administering the germs, disease causing germs, but the strength of that germs will be weak or they will be dead. So, during vaccinations, all this we have already done. All right. So, vaccination means the introduction of weakened germs or the dead germs into the body of an organism for developing resistance to a particular disease. So whenever we are administering vaccine into the body of an organism, that is called vaccination. And vaccination is simply done in order to develop immunity, and the development of immunity for a particular disease is called immunization. So all these things we have already discussed. Okay, so vaccines are available for a variety of diseases such as cholera, typhoid, measles, okay, chickenpox. On injecting them into the body, the person gets the disease in mild form, and his body gets stimulated to produce antitoxins, because whenever we are administering vaccine into the body, our body will produce the antibody against the toxins, or let's say against the pathogens that we are using during vaccines. Okay, this is the brief reason for you, because we have this already these things in the human health and disease chapter, in chapter immunity also. Okay, so it will be a brief reason for you. So during vaccination, we are giving the vaccines into the body of an organism, or the body of the organism will start developing immunity against the vaccine that is administered. Okay, now the antitoxin provide immunity against any further attack of the particular disease. How? Because vaccination is simply not only given to produce the immunity for a short period of time. During vaccination, one of the most important cell will be produced, which is called memory cell, and that memory cell will recognize whether the pathogen or the microorganism that will cause a particular disease is entering the body for the first time or the second time. If the pathogen is entering the body for the second time, memory cell will recognize it, and it will develop the antitoxins or the antibody against the said pathogen. Clear? So this is the most important principle of vaccination: development of memory cells. Clear? Now, two common vaccines obtained by growing bacteria are TAB vaccine for typhoid, where we use the killed bacteria, and BCG vaccines for tuberculosis, where we are using the living weakened bacteria. Okay, so today we have just simply discussed the use of bacteria in the production of medicine. In the next class, we will learn about the role of bacteria in agriculture. Okay, then we'll learn about the role of bacteria in industry also, and in the production of household products. Clear? So this much for today. See you in next class. Thank you.